So, hello, dear guests of Eternity Life Forum. Today, I will give you a brief introduction to the world of mitochondrial medicine and how mitochondria determine our health and lifespan. So, mitochondria are organelles inside our cells. Almost every our cell contains mitochondria. 2 billion years ago, they were bacteria and still have so many from bacterial origin. We all know mitochondria are the powerhouses of the cells, and they getting energy from food and converting it into the ATP, chemical energy, so we can use it for our chemical processes within cells. But they have many other essential functions: immune function, signaling function, synthetic, apoptotic. So now we tell mitochondria are bioenergetic and biosynthetic signaling center and not just powerhouse of the cell. What happens when mitochondria become dysfunctional? When mitochondria become dysfunctional and do not work appropriately anymore, they are making less energy in ATP form, and we all feeling it when we age. We are not that energetic as we used to be. They generate also too many reactive oxygen species, which damage our cells, and also there is an onset of sterile inflammation because of mitochondrial dysfunction. Why? Remember, they were bacteria, and mitochondria are hidden from our immune system with two membranes. And when they are damaged, when membrane is damaged and mitochondrial DNA or lipids from inner mitochondria membrane came out from mitochondria, our organisms just think that's bacteria and start inflammation and inflammaging as a result. So many things that damage mitochondria. When we eat too much, infections, different medicine, genetic predisposition, sedentary lifestyle, our gut microbiome, deficit of vitamins and trace elements, air pollution, and age, all these factors are leading to mitochondrial dysfunction. You may think mitochondria are something tiny which are not related indeed to health, but if your weight is 60 kilos, the weight of your mitochondria is 6 kilos, so 10% of you are mitochondria. Different cells have different amount of mitochondria within. So the richest cell with mitochondria are oocytes, women germ cells. Then neurons have a lot of mitochondria, cardiomyocytes, hepatocytes. And look at which systems of our body age first: brain, heart, muscles, sarcopenia, fatty liver disease, liver, yes, and also a women's reproductive system. As you know, women's reproductive aging begins earlier than aging of all other systems. So mitochondria rich tissues age first. Is it a coincidence? Not, I don't think so. So let's go further. Mitochondrial dysfunction leads to aging and age-related diseases. All of them, actually. Mitochondrial quality define cell stemness. If something is wrong with mitochondria within stem cells, it can't be a stem cell anymore. It's starting differentiating. Mitochondria determine also cell senescence, because if there is lack of energy, bioenergetic capacity of cell decline, and cell can't anymore pack proteins appropriately, etc. Mitochondrial dysfunction, as we have already mentioned, is leading to sterile inflammation and immune aging. Mitochondrial dysfunction is a root of all metabolic diseases and sarcopenia, since mitochondria can't work with food, can't process nutrients as they were when they were young. And also mitochondria in muscle decline lead to sarcopenia. Mitochondria decline lead to cardiovascular diseases. Then again, energetic function and function of calcium buffering, which are related to mitochondria. Mitochondria dysfunction also leads to cognitive decline and all of the neurological diseases. So aging and all age-related dysfunction is so interconnected with mitochondrial dysfunction. So what is mitochondrial medicine? So many things that damage mitochondria, but how to fix our mitochondria, how to push them to work better? Mitochondrial medicine is a set of measures aimed at improving mitochondrial function, not only in mitochondrial inherited diseases as it was before, but also to prevent development and to treat age-related diseases. Because now we know that mitochondria are a keystone of aging. Actually, there are three directions in mitochondrial medicine: diagnostics of mitochondrial dysfunction, functional approach, kind of lifestyle interventions, yes, and mitochondrial therapy, high-tech approach. Okay, how to measure mitochondrial functions? Let's start with diagnostics. Since if we do not have biomarkers, how can we talk about improvement of mitochondrial functions? Actually, mitochondrial diagnostics is quite tricky, and still there are so few reliable biomarkers, so we need to combine some of them to get some results. Why? Since there is a huge tissue specificity in mitochondria. Mitochondria in heart, for example, may be still okay, but in muscle has already declined, or in brain has already declined. Then we have too many mitochondria in one cell, and some of them can be damaged and some of them functioning good. So there are many issues in mitochondrial diagnostics. And we usually combine three approaches to mitochondrial diagnostics. The first one is functional testing. It's provided by measuring VO2max and other parameters. Sometimes lactate is taken at the beginning of exercise when there is a peak effort and there is a renovation phase. It's a very good approach. It's good indeed because it reflects mitochondria functioning in the whole of your body, but it needs special equipment. Blood tests are very convenient, but a single reliable biomarker does not exist, so we need to combine and test a set of biomarkers, and we'll talk a little bit about it. And genetical testing we do it usually to find a mitochondrial mutation for primary mitochondrial diseases, but also we can use it to measure mitochondrial amount in your cells. So let's start with biochemical biomarkers. We'll not go into too much details, but when we can measure actually some deficits that don't let mitochondria to function appropriately. So which are the best studied and available? It's we can estimate glutathione status, so we can test in blood reduced glutathione or just a ratio of reduced and oxidized glutathione. We can test vitamins needed for mitochondria functioning, and if we have deficit, our mitochondria can't work well. We can test as well trace elements which are essential for mitochondria functioning. Then among biochemical biomarkers, organic acid test is very popular, and it's a powerful tool to analyze mitochondrial function and deficits as well. Usually it's provided with urine testing, not blood, and it's very convenient, but it needs proficient interpretation and also high quality laboratory equipment. So the principle of this test, it is quite easy. You see different metabolites, how do they convert within mitochondria and form a cycle, a Krebs cycle. And to convert them one into another, we need special enzymes to function appropriately. And if some of the enzymes is not functioning as good as it has to be, you will see that one metabolite will accumulate in high amount and cycle will not work appropriately. So it can happen due to the deficit of, for example, cofactor for this enzyme. And then if we found that, for example, this enzyme is not working as good as it used to be, and its cofactor is, for example, B2, we can just use B2 to improve this enzyme functioning. So what we can test using organic acid test? A mitochondrial function, different steps of metabolism of neurotransmitters, cofactor deficiencies, and detoxification indicators. Then biochemical markers, oxidative stress. We will talk about oxidative stress a little bit later, but there are some markers that show us the damage is already occurring and our molecules like lipids, DNA are already under free radical attack. So we can measure whether lipid oxidation is ongoing, protein oxidation is ongoing, or DNA oxidation is ongoing. There are specified biomarkers and we can test them with blood test. Genetical markers. As I have already told, genetical markers are widely used for clinical diagnostics of primal mitochondrial dysfunction inherited. But we can also use the test determining mitochondrial amount, and this is a marker allowing us to estimate how many mitochondria we have. So mitochondria quantity, not quality. And it's very useful when we do some interventions aimed on increasing the mitochondria amount. So we can measure this marker before the intervention and after and see whether the amount of our mitochondria increase. So how can we improve our mitochondria? And even so technologies are developing still, nothing is yet better than physical activity. Physical activity increases the number of mitochondria and makes them more efficient. 1 month of physical training increases mitochondria content up to 100% and its bulk density, so it functions better by 40%. It's shown to be effective for insulin resistance and sarcopenia treatment and prevention. And actually, physical activity affects mitochondrial biogenesis not only in muscles, but also in our brain. And it was shown that 30 minutes of physical activity per day increases the amount of specific RNA, which is responsible for memory processes. So we're doing our memory better when we exercise. But the question is, what is the minimum amount of physical activity to boost our mitochondria? Actually, studies show that everything is good. No matter what you choose, moderate training, high intensity or super high intensity training like sprint interval training, everything doing mitochondria better. It stimulates mitochondrial biogenesis and mitochondrial work. The only thing is moderate continuous intensity training, like 4 hours per week. It mostly increases mitochondria amount, and high intensity training, it more doing existing mitochondria to function better, but not increasing the amount of mitochondria. But then again, everything is good, whatever you choose. The second pillar of mitochondria functioning is nutrition. We just can't go further without appropriate nutrition. So many things about it is crucial, but I want to focus your attention on few of them. Switching between substrates, it's what we call now metabolic flexibility. When we eat too much, it's bad. Hunger, not continuous, but time to time hunger stimulates mitophagy and biogenesis, so make our mitochondria better. Fatty acids are very important for mitochondria functioning. We need to supply our mitochondria with appropriate amounts of needed vitamins and trace elements. And when we food our cells, we also food our microbes, and they do affect mitochondria functions. So the first thing, which is very, very important, a little bit of theory, and you will see why metabolic flexibility is so important. So actually, we get our energy for 2 main food sources, from fat and from carbs. Yeah. The food excess is always stored like fat in fat depots. Carbs proceeds without our gut and gives glucose as a simple compound, and fats gives us fatty acids. And then glucose turns into pyruvate and fatty acids turn into another compound. So carbs and fats breaking down during digestions and form small molecules. And they go into mitochondria to give us energy, but do it in a slightly different way. So, have you ever been in a queue to a doctor? I hope so. Imagine the mitochondria is a doctor within the cell office, yes? And fatty acids, which came from fat we consumed or from fat from our body, which was stored in our body, they are patients standing in a line to oxidation in mitochondria to give us energy. But glucose never behaves like that. It has insulin. Insulin, you have heard about it for sure. That pushes glucose into the medical office out of a queue through a side door. So it pushes it through a specific glucose transporter. So when we have glucose and insulin levels high, they will always go first. When we are not eating for some prolonged time, like night period, and when we do exercises, glucose is a little bit depleted and its level decline. And fatty acids standing in a queue can come into mitochondria to be oxidized. And when we visit our refrigerator every 2 hours, fatty acids wait forever and accumulate. When they are accumulating, they try to force mitochondria and mitos coming into being overloaded with substrate and oxidize fats just partly, leading to accumulation of ceramides and other lipotoxic products. And from the other hand, since fats trying to not to let glucose into mitochondria, yes, because they were waiting too long, and when they accumulate, they're just trying to force mitochondria to oxidize them, not glucose. And glucose needs more and more insulin to be pushed into the cell, and that leads to the insulin resistance. But fats are also important for other things. Because fatty acids, they are a material for building inner mitochondrial membrane, and we need a lot of fatty acids to build our mitochondria. And fatty acids are really important for brain functioning because actually there is, you may have heard, our brain is just glucose. That's not true. When the glucose is depleted, our liver make ketone bodies which fuel neurons. And moreover, ketone bodies have important signal functioning. They stimulate neurotrophic factor, yes, BDNF, which promotes synaptic plasticity and neuron growth. And ketone bodies, especially beta-hydroxybutyrate, is a good one and has so many important signaling functions. So it's very important to be metabolically flexible, deplete a little bit our glucose and then oxidize fats and then go back to glucose and then again oxidize fats. So what is better, fats or carbs? And that's a very important and very painful question for all the people. Actually, the switching between them is the best. It stimulates both stress resistance and plasticity pathways. So how much is enough to keep your metabolic flexibility in form? Actually, 10 to 14 hours of hunger, or for example, 1 hour moderate training after 4 hours without food. Another important stuff of food we often not think about that we eat. Then when we eat, we feed not just our mitochondria, but also our gut microbiota. And different metabolites from gut microbes affect mitochondria in a large extent. And one of them, which is famous and everyone is talking about now, is urolithin A. Relatin A is a postbiotic. It is made by our gut microbiota from ellagic acid and ellagitannins. Ellagic acid and ellagitannins are contained in different plant food, like grapes, strawberry, onion, nuts. But we need to have special microbiota to do it. And relatin A was shown to induce mitophagy. Mitophagy is clearance of bad old mitochondria which generate reactive oxygen species. So when we delete it, our new mitochondria can divide and so to renovate pool of our mitochondria. And it's now under clinical studies, and it has shown some really good results. Human clinical trials have shown that relatin A supplements, so we can get it from our microbes, and also there are supplements made for people, for example, who don't have appropriate microbiota, have shown to increase indicators of muscle strength for kind of around 15% for elderly people without any other interventions. It's indeed a very good results. Other things besides calories we are getting from food are nutrients. Mitochondrial work is a conveyor, and if something is not working appropriately, something gets stuck, everything is out of order. So be sure you have taken enough and don't have deficits of vitamins. The essential are group B vitamins, vitamin C and vitamin E. Trace elements, especially zinc, selenium, and magnesium, and some other components. Mitochondrial medicine for many people is clearly associated with the intake of antioxidants. So I cannot say a few words about them. First of all, they are so different. When we talk antioxidant, we should mention that which exact group this antioxidant belongs to. The first one is one that directly neutralizes reactive oxygen species by binding to them, like vitamins A, E, glutathione, melatonin. But there are another antioxidants which actually can boost your natural antioxidant defense systems, and they work the best. You can see the list of them. Melatonin is also working in this group as well. And there are antioxidants that can reduce electron leakage and reactive oxygen species generation, but by improving mitochondrial functioning. So a little bit of theory about oxidative stress. Actually, reactive oxygen species and energy are two sides of the same coin. How is the energy generated? The food we eat from small molecules, and after all the biochemical processes, forms the simplest atom, hydrogen, which goes into mitochondria. Then it breaks into the electron and proton. Proton goes into the intermembrane space, and it's generating a kind of a gradient like an accumulator. And now mitochondria like an accumulator. In the inner mitochondrial membrane, there is a special protein complex which is called ATF synthase. It's the nanovehicle. And when the protons moves from the intramembrane space again into the mitochondria, the energy from a gradient to ATP form is accumulated. So actually it's similar to the water in the water tower. Yes, you put the water upside, and when it goes down, it makes energy. But overcharging the membrane is bad. If we have too much protons in the intermembrane space, when we eat too much, for example, the ATP formation speed is not that high, and it may lead to the oxidative stress, since electrons into the electron transport chain, they can just jump out from that and form reactive oxygen species, damaging DNA molecules, lipids, etc. So actually, nature have discovered a natural protective mechanism from it, which is called uncoupling, and it is made by uncoupling proteins. There are special protein channels which just let protons go inside mitochondria, and energy is dissipated as heat without ATP generation. So we don't get energy, but we are not getting oxidative stress as well. A lot of UCP proteins making uncoupling contain mitochondria of brown fat, which is a good fat, which burns fat, fat that burns fat, yes. And all the energy stored in fat just dissipated as heat. The bears have a lot of such fat, and that's why they are metabolically healthy, even they are fat. And the formed fat allows them to survive sleeping during the winter. And some of our white fat cells, which are metabolically not always healthy, probably can go brown and can help us reduce fat amount and not to form free radicals. And there are many approaches how to make our fat to be brown. The most famous is cold exposure, sleeping in cold places, for example. Taking some hot compounds like capsaicins. Ketones and lipids also stimulate a little bit fat browning, and some drugs like sildenafil, Viagra also stimulate fat browning. And also there are a lot of different approaches which are tested on animals, like gene therapy or optoelectric stimulation of neurons. So everyone want to have healthy fat and eat without getting fat, kind of that stuff. So then again, you may never think about reactive oxygen species are not always bad. Actually, ROS are only signaling molecules that tell our organisms and our cells that something goes wrong with mitochondria. It's like a temperature when you have a flu, yes? When it is not too high, it helps our immunity to fight microbes. But when it's too high, it may cause damage. The same stuff is going with antioxidants and free radicals. So free radicals are not bad themselves, but when our adaptive and antioxidant systems are depleted, they accumulate in too large extent and can lead to damage. And antioxidants do not eliminate the cause of dysfunction, and they do not affect the functioning of respiratory chain, but sometimes it can be useful when oxidative stress is already exists, and we can see it from the markers of oxidative stress we have already mentioned. So it's not a magic wand, and for healthy people, antioxidants taking is actually really limited, and for spot activity, and even can reduce the markers of adaptability. Among antioxidants, I would mention melatonin. It's the oldest antioxidant. You may know it as the sleeping hormone, and actually it's not only. It's a powerful antioxidant, and mitochondria have the ability to synthesize melatonin, and its level drops with age, and it works really good as antioxidant. Another one, which you can hear about, is NAD+ precursors. NAD is a centric metabolic factor that plays an essential role in regulation of cellular metabolism and bioenergetics. And a lot of decisions cells are making based on the changing of ratio of oxidized and reduced form, like cell senescence. And supplements of NAD+ donors have really good results on rodent models, improving mitochondrial function and all other functioning. In human clinical trials, NAD supplementation didn't show that good results. It can improve a little bit mitochondrial function in combinations, but then again, it's not a magic wand. And you can boost your NAD+ synthesis by doing exercise, by doing calorie restriction, etc. The other stuff by which we can improve our mitochondria love us happy, it's serotonin actually. When there is a serotonin, it's a hormone, neuromediator of happiness, and when someone tells us we are good, and it acts through its receptor and it stimulates mitochondria biogenesis in our brains. So social networking is so important. And it was also shown that agonists like amphetamine can also improve mitochondrial biogenesis, but this work was done only on cells, so I would not go into it too much. Another stuff is nitrogen sulfide. It's a terribly smelling gas from rotten eggs, which can also affect mitochondrial functioning. We get it from two sources, from our gut microbiota and from our own metabolic pathways. And hydrogen sulfide from our metabolic pathways is essential for mitochondria functioning, but it has to be in the optimal amount, not too high and not too low. And the things it's made in a reaction with CBS enzyme. And the things you should keep in mind that when you have high level of homocysteine, it inhibits the formation of hydrogen sulfide. And also synthesis reactions cannot proceed normally without a sufficient amount of vitamin B6. And also drugs which are donors of hydrogen sulfide are developing, but probably not everyone needs them. But for people with, like for CBS polymorphism, they can be useful. There are also some innovative approaches are developing, like mitochondrial gene therapy, transfer of mitochondrial genes, broken one maybe, into the nucleus, mitochondrial transfer on transplantation, or mitochondrial augmentation therapy. So when we put healthy mitochondria from our healthy tissues, for example, or from donors into our cells. I would a little bit talk about mitochondrial transplantation. I've been recently at Mitochondria World Targeting Congress, and there were so many reports about mitochondrial transplantation. So when we take mitochondria from our own tissues like muscles or from donor tissues, isolate them and then inject mitochondria into the bloodstream or into the target organs like heart, like liver, or even brain in animal models. And this mitochondria then boost our cell functioning. And actually, autologous mitochondrial transplantation is in clinical trials for stroke treatment, so maybe we will have some good results soon. Another approach is low-level laser therapy, very interesting approach by affecting our mitochondria not through chemical compounds, but through physical forces, yes. The laser of specific wavelengths can affect cytochrome oxidase within mitochondria and affect mitochondria functioning, like increase ATP level, decrease ROS level, etc. And low-level laser therapy for cognitive impairment through applying specific devices into our corp is developing, and it's really safe and a good approach. So thank you very much for your attention. I hope it was interesting, and wait for your questions. You can as well write me into email or into Viber, WhatsApp, whatever. I will be happy to answer. Thank you.